This picture is courtesy of Kennebacus Valley High School and shows the stages of mitosis. Before interphase occurs, there are three things that need to happen. The first is protein synthesis. The cell needs to be able to synthesize all its proteins and enzymes in order to grow and copy. Then is ATP production. The cell needs to generate enough ATP, or cellular energy, to divide. Then they must increase all of its numbers of organelles so that each daughter cell will have enough organelles to survive. Interphase is made of three different parts. G1, where the cell grows and reproduces its organelles and proteins. G2, where the cell grows even more and prepares to divide. And the S phase, where DNA replication occurs. A new DNA is synthesized. This is also where the chromosomes are copied. Prophase is the first step in mitosis, and this is where the chromatin in the nucleus begins to condense into chromosomes, which then become sister chromatids. This means that the DNA on each chromatid is identical, so that both daughter cells will have the adequate information to survive on its own. Then the nucleolus disappears. The centrosomes release fiber and centrioles, which move to the ends of the cell and extend towards the junction of the sister chromatids. These fibers create the mitotic spindle. Prometaphase occurs in between of prophase and metaphase, and it is where the nuclear membrane dissolves and proteins attach to the centromeres. This makes them kinetochores. Then the fibers of the mitotic spindle connect to the kinetochore. Here the goal is to ultimately pull the sister chromatids apart and then pull them to the two daughter cells. Metaphase is the next step in the mitotic process, and this is where the spindle fibers pull the sister chromatids into the middle of the cell and then line them up along the metaphase plate. This ensures that new nuclei will receive one copy of each chromosome during anaphase. Anaphase comes next, and this is where the sister chromatids finally separate, but they remain in their condensed form. Then they move to opposite sides of the cell due to the action of the spindle fibers retracting to the centrioles. By the point that telophase begins, the sister chromatids are on opposite sides of the cell. The nuclear vesicles will now begin to form around each group of chromatids. As this envelope forms, the two nuclei form within each of the cells. Then the microtubules and the kinetochores dissolve and the chromatids begin to decondense and diffuse back into chromosomes. The last phase in mitosis is cytokinesis, and it varies for plants and animals. We'll start with animal cells. A fiber ring of actin contracts around the perimeter of the cell. This separates the two cells completely and effectively creates two identical daughter cells. In plant cells, they take this one step further by synthesizing a cell plate between the two daughter cells. This is what forms the rigid cell wall of cellulose and therefore creates the two cells. The first and potentially most important reason that mitosis is important is that it allows for the growth of multicellular organisms. The means to grow is to increase the number of total cells, and this can happen only through cell division, aka mitosis. This growth is also helpful in dealing with tissue repair in multicellular organisms. Damaged tissue can be repaired by replacing damaged cells with new and identical ones, created by mitosis. Asexual reproduction is also an important use of mitosis because it allows for unicellular eukaryotes and prokaryotes to reproduce and pass on their genetic material. During mitosis, one cell divides into two genetically identical cells. Some organisms, such as protozoans, reproduce this way.
Tumors are the result of uncontrolled cell division, and there only needs to be one cancerous cell to infect the entire system. Cancerous cells are dangerous because they are unable to regulate cell division and therefore reproduce uncontrollably. This tumor can then be spread into various parts of the body and cause major illnesses.